For today's webinar, I will go through some of the background of how the pollen tube growth model was developed, how it's used, and then I'm going to pass it off in a little bit to Dan Olmsted, who is the director of the NUA system at Cornell, which is how most of the people in the eastern United States are going to be accessing the pollen tube growth model. And he'll do a run through about how to actually, the practicalities of actually running the model on that platform. If you see on the screen here, this work while I'm presenting today is actually the work of many, many people, in particular my Virginia Tech colleagues, Dr. Keith Yoder, Leon Combs, who was Keith's technician for many years and actually did most of the nitty-gritty lab work to create these models. Candace DeLong, who was a master's student with me a number of years ago when I was at Virginia Tech. And actually not on this screen is Ross Byers, who actually came up with this idea originally probably more than 15 years ago, about how we can use pollen tube growth rates to time thinning sprays better in orchards. At Cornell, a number of colleagues, including folks who are on the call today and others who have either worked with me on beta testing and or getting the model implemented into the newest system. So there's a number of other reasons why people have issues with bloom thinning, but I guess spring frost is often one that comes up, and the fear of trying to do some bloom thinning and then a few days later having some sort of frost event and reducing the potential crop load even more so. Lack of empirical data and experience, the timing of the bloom sprays, and that's really the key aspect of the pollen tube growth model. It's really about when to put on the applications. That's the primary thing that the bloom thinning, or that the pollen tube model does. And then lack of materials that don't have side effects. And I'll cover a little bit of that. I've had some conversations with a number of the different companies that are making products that we found to be efficacious for bloom thinning, and give you some updates on that. The pros and the reason why bloom thinning is really good is because and we've known this for decades in pomology, is that the earlier in the season that you reduce the potential crop load, the larger the fruit size is going to be and the greater the return bloom the following year. And so, and that's because as soon as you have pollination and fertilization and seeds start to form in the fruitlets, they start producing plant hormones, gibberellins, auxins, etc., and those compounds will actually inhibit flower bud initiation. So the earlier you get those, you prevent those flowers from turning into fruit, the more likely you are to have flower buds to be initiated for the following year. We've done some work looking at some of these materials because they tend to be caustic, and they originally were developed as fungicides. So if we were to overlay their use as the bloom thinner with their use as a fungicide, when can that work? And that's something we published a paper on that, and I'll go over that a little bit later today, which helps to reduce the number of applications that you might have to put on in a given year, and particularly for organic growers, since these materials tend to be organically approved, that's a real option. And then the pollen tube growth model, which we'll talk more about, helps to have more precise timing for those applications. So just a little reminder on flower anatomy, and what we're talking about is, remember that this is the green part here is the female organs within the flower. And the top here is the stigma. That's where the pollen grains land. When they land there, there's a number of different things that happen. The pollen grain will get hydrated. It gets some stimulation from plant hormones that are in the stigma, and then that pollen tube will start to grow down the style. That's this long green stalk here. And as that pollen tube grows down, it eventually grows into the ovule, and inside of the ovule is where the egg is, and that's where fertilization happens. So today when I talk about fertilization or it's being fertilized, I'm talking about the reproductive event. I'm not talking about adding nutrition to the orchard. And then of course this is the male part of the flower here. This is the filament. That's the stalk on the male side, and then this is the anther up top. That's where the pollen is located. So the model was developed by researchers at Virginia Tech for over the course of many years, and we actually came up with a pretty cool system using root bag trees, which allow us to grow the trees in the field. Let's see if I can move this over a little bit. Allows us to, in the top right corner there, to grow the trees in the field inside of these kind of almost like burlap sacks, or a little different than that, but kind of like that. Dig them out in the winter time, and we'd actually some years put them into refrigerated trucks so they could accumulate their winter chill units, and then pull them out of the, once they'd accumulate enough winter chill units to break dormancy, put them into a greenhouse so that they can start to grow and flower. And then we do controlled pollination. Here you can see on each of these trees a number of different colored tags. Each of those was, in this experiment, different pollen sources. We're looking at what the effect was of different pollen sources on the rate of growth of the pollen tubes. And then we can put them into growth chambers. So that's what you're seeing in the bottom middle picture there. And those are two trees inside of a growth chamber. Growth chamber is basically allows us to have control of the temperature, the humidity, other factors, the light levels as well, to be able to do replicated experiments. And so in the buckets there, that's in a greenhouse, we do the controlled pollination. We actually emasculate the flowers first so you don't get any contamination. And then you do your controlled pollination. Once you pollinate the flowers, you put them inside the growth chamber. Usually, most of the time we're doing these for 24 hours. And then after 24 hours, you pull them out of the growth chamber. You can remove the flower at that point. We'd preserve it into a solution. And then when we're ready to do the evaluation of it, we would add a dye that would highlight the color of the pollen tube. And then we can use on the software that comes with a lot of microscopes, be able to measure the length of that pollen tube. And so through this system, we're able to run the growth rate of the pollen tube at different temperatures. So we'd be able to then, by doing this multiple times, number of replications, be able to build the model for at different temperatures, how fast is the pollen tube growing. So just some more pictures along the top there, some that you've seen. The brush picture there is actually, you can see that that flower has been emasculated, and that's actually pollen on the end of that brush that we're doing a controlled pollination. Very similar technique to what apple breeders will do. And then on the far right on the top, you'll see this is what the pollen tubes look like, and they're highlighted in blue with the dye there. There's some pubescence at the bottom there. We're not really looking at that, but you can see the blue arrow at the darker blue arrow at the bottom. And you can see how we measure the pollen tube. So this is 8.3 millimeters, for example. And so on the bottom left, you can see the on the x-axis going across on the bottom, temperatures in degrees Fahrenheit. And then on the y-axis, this is the actual growth rate in millimeters per hour. And so at different temperatures, you can see that we get different growth rates. And so they tend to be very slow as you're cold, right? And then as temperatures warm up, the pollen tube growth rate increases. So that's fairly intuitive. And you can see at some point in here, it's just a little bit shy of 80 degrees Fahrenheit, the growth rate tends to slow down. And this is really common in a lot of biological systems, is that there's kind of an ideal temperature. And then at some point it gets too hot and growth rate actually starts to slow down. And so the ideal growth rate for Fuji would be about 80 degrees, just shy of 80 degrees, for example. And so you can see here, this is how these models were developed, and we had to do this over and over again for each of the different varieties that we were working with, because there were enough differences among different varieties. And the Fuji would be the maternal variety, not the pollen source, but rather the variety of apple that you're growing in your orchard. Okay, so then on the right-hand side here, how do we translate that to something practical, something that growers can use? Because people like me get really excited by the physiology of this, and it's kind of cool to just measure pollen tube growth rates, and I'm generally interested in that kind of thing. But for a practical use, how do you translate this into something that can be used for crop load management? So the way this works is on the bottom here, we'll start there, is temperature. And you can see this is diurnal temperatures. So this is the high temperature during the day, low temperature at night. And you can see it kind of swinging up and down over the course of several days here. And so that then gets translated into the pollen tube growth rate. So this is what the model looks like when it's implemented into the NUA website. And so as temperatures are on the lower end, a bit cooler, you can see growth rates are slow. And then you can see as we get across here, as temperatures start to warm up, you can see the growth rate increases here. Okay, so it's using the equation from the graph that you see on the left-hand side to translate the temperatures that you see on the bottom into the pollen tube growth rates that you see in the top. Okay, so some interesting things here we need to think about. One is, when do you start the clock? When do you start the model? Okay, so that's where the little red pointer is. When do you put your application spray on? Okay, so this would be the first spray here. And then the clock automatically resets to 0 and runs again. When do you put that second spray on? Do you need a third spray? Okay, so these are some of the things to know. And then so this is the x-axis is day and time. And then on the vertical axis, or the y-axis, is the percent of the style length. Okay, so we use style length to calculate when to start the model or when to stop the model. So style length then again is from the top of the stigma, if you look on the top right, to the bottom of the style. Okay, and this is our style length. Okay, so this is our starting point. This is where the race starts, the stigma. This is the finish line at the bottom. Okay, so we're going to assume the finish line is fertilization, right? So pollination and then fertilization. And so what we're watching here then is we're assuming this is when, at the start on May 5th at 12 o'clock, this is when we started the model. So we assumed at this point there were enough flowers open. We're usually thinking about king bloom flowers. There are enough of them open on the tree or in the orchard to start the clock. Okay, so this is when the last flower that we want to turn into an apple for the growing season is open and receptive to pollen. And then we start the clock, and it's using these calculations on that model on the left. And then this is 100% here. It's got up to 111%. When we get to 100%, that's when the pollen tube length is equal to the style length. Okay, because at that point, we're assuming that that last flower that we wanted to be part of our crop for that year is going to be fertilized and turn into an apple. At that point, we do not want any more flowers to turn into apples. So we put on some sort of thinning spray. Typically, we're using something caustic, lime sulfur, ammonium thiosulfate, or ATS. We've found some good use with a Marone Bioscience biofungicide called Regalia, which is a knotweed extract. And then we allow another wave of flowers to get pollinated after that caustic thinner went on. Those pollen tubes are growing. But at this point, we do not want those pollen tubes to reach the length of the style, because we do not want any of these flowers that are open at this point in time, May 9th at whatever time that is, 3 o'clock or so. We don't want any of those flowers to turn into fruit. So we don't allow them to reach the base of the style and fertilize the flowers. So that's why we stop it at 70%.
Hey, Greg, can I ask you a question from Chris?
Yeah, go ahead.
So Chris is wondering, do you start when you have enough open flowers or when you have enough open flowers during decent pollination weather? How much time of good pollination weather is necessary before you can assume you've got pollen on the king blooms?
Great question. Okay, so, and we'll talk a little bit about this when I go into some of the research that we have done as well. But we know that our European honeybees tend to be fussy and they don't really fly that much below 55 degrees, and typically they like temperatures in the 60s and 70s and above to really have good flight. They don't like to fly when it's raining out either. Some of our native pollinators tend to be a little less fussy and they might be out and about. That really depends on your specific conditions in your orchard. So yes, the model is assuming good pollination weather and adequate pollen availability and good flight by pollinators. And when those factors are not met, it's something to weigh as to how many flowers you want open to start the model clock. And at that point, we don't really have a good model for saying, how much more time do you need? I think that is something that an area of that'd be great to have some additional research in, but it really comes down to the manager of the orchard to use their intuition and knowledge to say, I think we have good pollination.
In your experience, what have you found enough time to be? A day with good king bloom open and bee activity?
Yes. When conditions are right and the bees are flying and you have adequate pollinizers in your orchard, either other varieties or crabapple trees, usually within a few hours they're out there. And if your flowers are open and receptive, they often get pollinated within just a few hours. Okay, so this slide is just showing the same in a larger scale, and I think I may have just kind of went ahead and talked about most of this already in the previous slide. And it's the same data again. So the model clock started at May 5th, 12 o'clock. These are the pollen tubes growing up to the style, and it's 111% the length of the style here. We put on the caustic thinning spray. We start the clock back to 0. Pollen tubes are again growing to the base of the style. We put on the application at 70% the length of the style because we don't want these to be fertilized. We wanted these flowers here and that first curve to get fertilized. We do not want these to be. And then I don't think this is just the end run of the model here. In most years, in our conditions in New York State and the work I did at Virginia Tech, we found that 2 applications was often adequate for achieving acceptable thinning. In Washington State and other parts of the Western US, I've heard of growers typically putting on 3, sometimes even 4 applications in a year if they have a really big bloom or if they have a lot of rat tail bloom they're trying to get rid of. Okay, so there are some parts of this that the growers that you'll need to do, right? It's not a fully automated system. It does require some level of work on your behalf to figure this all out. And so the first thing you have to do is for every variety that you want to thin, you actually have to measure the style length. And the picture in the center of the slide there shows what we're talking about, the top of the stigma to the base of the style, and that is the that is the length of the style. And let me see if I can get out of here for a second. And I have a short little video from Keith Yoder, my colleague at Virginia Tech. Keith has done a lot of work to really push this model forward. So I'll just show a couple of these slides here. And so the King Bloom style samples should be taken as early as possible to establish the average style length. And so really you are going to be taking the those measurements as soon as the first King Blooms open. And that should give you typically between 2 and 5 days before you put on your application of your first thinning spray. Okay, so there is enough variability among varieties and even within one variety from one year to the next that we recommend measuring this again every single year on every single variety that you want to use the pollen tube growth model on. We find that somewhere between 25 and 50 flowers is what's needed to get a robust mean of the style length for each block. And then as we showed you in the previous picture, there's a number of different styles within each of the flowers, each individual flower, and what you want is the longest style from each flower. Okay, and so then you're going to add those up somewhere between 25 and 50. I'd recommend doing 50 if you haven't done this before. After you have a little bit of experience, you can go down to 25. And when Dan talks about the new application of the pollen tube model, he'll explain some of the cool features that we have for entering in this data right into the website. And so in some varieties, as shown above, the styles may be longer than the stamens. And others, the stamens may be longer than the style. So one of the fun things that you learn when you start doing this is how variable apple flowers are, and it's just cool and fun to look at. And so this is just showing, where you want to measure to the bottom. The top is pretty easy. It's pretty easy to see where the top of the stigma is on top of the style, but at the bottom it can be confusing. And so if you can see the red line on the right-hand side there, that's basically if you push down the petals and then kind of down to the base of the calyx there, that's what you're looking at. And that's the length that you want to measure. And that's really important because we're talking about an overall length of just a couple centimeters, or often even less. And so, small variability in how you're measuring the bottom of the style can affect your results. And so here's when clearing away the filament and the anther, so you can just see the style, and that's kind of where the base is, where you want to be measuring to the bottom of. And you can use a ruler. We typically use calipers in my lab to measure this. It sounds like a lot at first, but you'll find that once you've done this a few times, it actually goes very quickly. And so it can be either be done on the tree, or you can actually remove the flowers and do this off of the tree. I typically prefer to do it off of the tree. I just find it to be a little easier to go out with a Ziploc bag, pre-label it, go collect my flowers, go back to somewhere where it's more conducive to focusing, and have my calipers there, have a piece of paper with me, or go right into the NUA website and enter in the data. And that's how it looks on the AgWeatherNet. So I'm going to stop sharing this screen now and go back to my PowerPoint. Okay. So just to review, I mentioned this previously, but starting the model clock is one of the more difficult aspects of this because it requires some user experience and expertise. You need to have sufficient King Bloom open, because those King Blooms are what you want to be your desired crop load for that year. Some ways of doing that is you can actually do counting flowers per branch or tree cross-sectional area. So if you knew you wanted, 6 fruit per cross-sectional area, you can actually count that out. You can measure it and count it. And I'd say even, just 3-5 years ago, that seemed to be a major barrier for a lot of growers. And I think with work that pomologists are doing in New York and Michigan and Massachusetts, around the country, really, growers are more comfortable with and more used to needing to count to get their desired crop load. And so that also is a part of this model. Sometimes growers just have a good sense for it. You can drive through an orchard and figure out when enough flowers are open. But if this is the first time doing it, I definitely recommend counting. And again, the model starts when the last flower that you need to achieve the desired crop load has been pollinated is open. Okay, and the first thinning spray is when the is applied when the pollen tube growth has been modeled to grow beyond the longest style, right? So 100%, and then usually a tad more, like I showed you that that graph previously that showed 111%. And then additional thinning sprays are put on to prevent additional fertilization. And we already mentioned some of the conditions for bee flight, and also we just know that there's a lot of variability for when flowers open within a tree, within an orchard, high points versus low points in terms of the topography. And so these are just kind of some of the things that you have to account for and think about when you're using this model. So currently we have developed these empirical pollen tube growth models for 7 varieties that you see in the top left. These are chosen largely because Washington State Tree Fruit Research Commission paid for a lot of this research early on, and these were the varieties that they were focused on in Washington State. And so on the right there is how the model has been implemented in Washington State, and it's getting a lot of use there. I think bloom thinning has become one of the most common ways of crop load management in the state, in Washington State. They actually find more efficacy from bloom thinning than they do from 10 millimeter thinning using plant growth regulators, just because their climatic conditions are different than what we find in New York or other parts of the eastern US. And a little bit of the history of how the model was developed is there as well. But we're really excited by is started working with NUA in 2018 to get the model available for us in the eastern United States on our weather system. And last year we had it fully available on NUA, and it will be again this year. And so in a moment I'll pass it off to Dan, who will talk about how that's done. But I do want to point out, just some of the known limitations. We talked about bee activity, pollen availability, that kind of went over that already. We only have models for those 7 varieties right now. There's work underway for developing additional models, and also some work that we're trying to do in my lab to develop kind of a universal or generic model that may not be as precise as a cultivar-specific variety, cultivar-specific model, but one that could be used for, we just grow so many more different types of apple varieties here in the eastern US. Some questions about the efficacy or the mode of action of different bloom thinners. And I'll talk a little bit more about organic use of bloom thinning for organic orchards, which is really the only chemical thinning that's available for organic orchards right now, is to be bloom thinning, or if you're not using something very caustic later. So Dan, I'm going to pass it off to you. I'm going to stop my share. Dan and I published a paper in the Fruit Quarterly that's available for free now on their website about how the model is implemented. But Dan is going to talk right now about how the model works on NUA.